Yeah, thank you very much. My name is Kilian Eyerich. I'm dermatologist at the Karolinska Institute in Stockholm, Sweden, and I have the pleasure to share with you some insights on human models of atopic dermatitis. These are my conflicts of interest. If we talk about human models of in vivo models of atopic dermatitis, then we basically talk about the ATOPIC patch test. What is it? It is basically an epicutaneous provocation and challenge of allergens, which are typically mixed in Vaseline and are challenged just like a normal epicutaneous patch test for typically 48 hours on the back of a sensitized patient that suffers from atopic dermatitis. And in some of the patients, we elicit by using this specific allergen provocation an acute eczematous reaction that you can see on the right side. In the example here is a patch test to house dust mite. Well, this test was originally developed to be useful for daily clinical routine testing. And you see here in this overview that compares the performance of the ATOPIC patch test with a skin prick testing or serum measured IgE levels, that in fact the performance of the ATOPIC patch test might be a bit better than these two other methods. You see that, for example, the accuracy for cow's milk is around 85%. In general, dependent on the antigen and allergen, it is something between 65 and 80%. And with this, it's higher, in fact, than skin prick testing or IgE. But still, it's far away from 100%, obviously. And the main reason why it did not really make it into our daily clinical routine on a broad basis is that it needs careful standardization. This is just an old example from a publication. You see the same allergen tested here, house dust mite again, at several different regions of the body. At the left side is the back of the patient, and at the right side, you see the forearm. And you clearly see that the reactions are not at the same intensity level. So the ATOPIC patch test overall gives us quite a good feeling if a patient has a relevant sensitization against a given allergen. It is only or typically positive in patients with atopic dermatitis and not in non-atopic dermatitis patients. But the accuracy of the ATOPIC patch test is something between 65 and 80%, so it is not really very high. And in addition, it is quite an invasive procedure, takes a while, and needs careful standardization, which is why for the daily clinical routine, it did not enter the broad basis. However, we can now use this ATOPIC patch test as a model for several aspects of the pathogenesis of atopic dermatitis. And it's just one image to remind us again of this complex pathogenesis of atopic dermatitis and to illustrate which aspects of AD pathogenesis we might address and investigate using the ATOPIC patch test and which obviously will not work. And we all know that AD is basically based on three different factors, which is type 2 immunity, resulting in barrier damage, or the other way around, resulting in microbiome alteration, Staph aureus colonization, or the other way around. And it is critically influenced and triggered by several environmental factors and other factors of genetic predisposition, or, for example, innate immune activation, other cell types than type 2 inflammation. And especially this immune aspect can be addressed quite well as a human in vivo model atopic dermatitis, ATOPIC patch test setting, I'm sorry. For example, we do know that very early type 2 inflammatory events are critically mediating the eczematous response we do observe in the patients that clearly have a trigger of an allergen. For example, that clearly gets worse if they are in contact to house dust mite. We also do know there are subpopulations that react with a worsening of their atopic dermatitis when they get exposed to, for example, pollen allergens. And if we do that, and here you see again this patient that has been challenged with dermatophagoides, so house dust mite, in an atopy patch test setting, and we isolate and we take a biopsy from these atopy patch test reactions. And from there, we isolate the T cells that infiltrate the skin. What we do observe is a picture like this. Stimulated T cells from an atopy patch test reaction might secrete interleukin-4, which is the typical type 2 immune cells, interferon gamma, so type 1 immune cells, IL-22 or IL-17, so the typical T helper 17 cells. In other words, all these T cells infiltrate acute allergic reactions elicited by a specific allergen, in this case, house dust mite. But if we stimulate these T cells not unspecifically on plastic, but if we stimulate them with their cognate allergen, the allergen that elicited this inflammatory reaction, the picture is this. You see that virtually all cells that specifically recognize the allergen house dust mite here secrete interleukin-4, and they do not secrete interferon gamma nor IL-22 or interleukin-17. So by using this model of an acute and specifically induced eczematous reaction, we actually can prove that at least acute atopic dermatitis is mediated virtually only by type 2 immune cells. We can also use the atopy patch test to get some insights in the kinetics and differences in the ongoing reaction. And this is a very old publication that I show you here, which was basically the begin of the mystery of a switch in AD kinetics. What you see in the dashed line is interleukin-4 in an atopy patch test at 0, 24, and 48 hours. And you see a clear peak here at 24 hours. And then you see the solid line, which has the same kinetics, that is interferon gamma. And interferon gamma obviously comes a little later than interleukin-4. And that was when the switch of AD inflammatory infiltrate was born at early and acute type 2 phase, and the later dominated type 1 phase, type 3 or T helper 17 cells were not known at that time. Today, we do know that, in fact, at later time points, other T cells become more and more important than in the acute phase, but type 2 cells are dominant and important throughout the whole course, so we cannot really call it a switch any longer. A third example about how the atopy patch test can help us to understand the immune infiltrate in ongoing acute eczematous reactions is interleukin-17. Here you see an immunohistochemistry staining of an acute AD, and we do observe, in fact, IL-17 positive cells. So we and others tried and isolate them and characterize them, and we screened a lot of different T cell clones, and we identified clones which were specific for an eliciting antigen. In that case, it's again an atopy patch test to house dust mite. And they were able to secrete interleukin-4 and also interleukin-17. But when we stimulated these T cells with their allergen, we again only found interleukin-4 secretion, and they virtually did not produce interleukin-17, although we knew they were capable of it. And also the addition of other T cell relevant cytokines such as IL-23, IL-6, or IL-1 beta did not change that picture. But what actually did was the addition of bacterial-derived superantigens, such as staphylococcal enterotoxin B here, which is derived from Staph aureus. It stimulates the T cells, and here, in fact, it could induce interleukin-17 secretion. So that was an observation on plastic. And what we did then was to verify that in vivo in the human AD situation, again, using the atopy patch test setting, one time dermatophagoides alone and the other setting with staphylococcal enterotoxin B. And we isolated suction blister fluid and epidermal roofs and could in fact prove that in vivo interleukin-17 was induced by staphylococcal enterotoxin B. So in summary, the atopy patch test is a standardized model to induce acute eczematous reactions in sensitized AD patients. It never really made it broadly into clinical routine as it requires a high rate of standardization, and it gives us only an accuracy between 65 and 80%. but it can be used to investigate the pathogenesis of atopic dermatitis, especially the immune infiltrate. It mimics the early type 2 immune infiltration, but it also mimics the codominant immune infiltration that is observed in atopic dermatitis at later time points throughout the natural course of the disease. It is, on the other hand, clearly limited to investigate the pathogenesis, and it is always self-limited, maybe because there are no autoreactive T cells involved here, as it does not take into account any physical impact such as sweating or sun exposure, and it also does not take into account the microbiome. So it is not capable of fully displaying the complex pathogenesis of atopic dermatitis, but it is a highly useful model to investigate certain aspects of it. Thank you very much for your attention.